Good evening to everyone. Today we will learn something about the application of recombinant technology, especially in case of the animal uh, genetics and breeding is concerned. So this chapter is very important as it is one of the emerging topic. So the applications of the recombinant DNA technology are in different fields, including the animal breeding and also the vaccination productions and many more fields. So, what is this recombinant DNA technology? It is a pioneering work of Paul Berg, Herbert Boyer, then the Stanley Cohen in the early 1970s and led to the development of recombinant DNA technology, which has permitted biology to move from an exclusively analytical science to a synthetic one. That means it is rather than the laboratory oriented, it became one of the applicable science. So what's a recombinant technology? It is new combination of the unrelated genes that can be constructed in laboratory by applying the recombinant DNA technique. So these novel combinations can be cloned or amplified manifold by introducing them into suitable cells where they are replicated by DNA synthesizing machinery of the host. So then the inserted gene are often transcribed and translated into a new gene setting. So these genetic characters of the host can permanently be altered in a designable way which we are interested into it. So the recombinant DNA technology is used for many and different purposes of the research, then medicine, agriculture, and also the industry. The way in which the technology was developed was to re-engineer the microbial cells to produce foreign proteins. So the microbial cells are utilized in such a way that they are directed to produce the required proteins which we are intended for. Then human insulin is one of the first therapeutic proteins that was genetically cloned using the recombinant DNA technology. Then by 2001, over 80 recombinant DNA based products have been approved for treating diseases and also for vaccination. A further 350 recombinant DNA based drugs were being tested for safety and also the efficacy. Then, this technology is also an important tool in agriculture, being used to improve plants resistant to pests and to increase the crop yield. Then there is a different application of the recombinant DNA technology into the field of the engineering, genetic engineering, heredity, and also the genetics and breeding. And there are certain tests which developed for determining heredity disease and paternity, and diagnostic tests for hepatitis and human immunodeficiency virus, that is HIV, and production of the vaccines and protein therapies such as human insulin, interferon, and human growth hormone, and also the production of clotting factors for treating hemophilia and in the development of the gene therapy. Then another thing is the genetic mapping, linkage of the mapping, and gene mapping is a process of method of discovering the location of genes on a chromosome, and that disease transmitted from a parent to child is linked to one or more genes, and which chromosomes contains the gene precisely where the gene lies on that particular chromosome. So, the gene responsible for relatively rare single gene inherited disorders such as cystic fibrosis or Duchenne muscular dystrophy are also the one of the applications of gene mapping. And some of the diseases are also like this asthma, heart disease, diabetes, cancer, and psychiatric conditions. They can be mapped with the gene linking. And here you can see that genetic maps are the species specific and comprised of genomic markers and our genes and the genetic distance between each marker. So this can be effectively used for different biological purposes. So what's a gene therapy? And you might have heard that nowadays everything, including the cancer, the gene therapy is done. So here, disease caused by deficiency of certain gene product are amenable for replacement therapy. So this strategy is to clone gene into a vector that is taken up and incorporated into a genome of a host cell. The adenosine deaminase deficiency has been treated successfully with gene replacement therapy here. Then here you can see that a defective gene is replaced with a correct gene or corrected gene. Then that the disease can be treated effectively. For example, the sickle cell anemia is one of the diseases that can be treated using this technology. Then another disease, thalassemia, and various other metabolic diseases of gene therapy are under trial. Then the transgenic mice, etc., or transgenesis is another technology where in which the laboratory animal are being produced. For example, the transgenic mice are one of the important things here. Then the somatic gene replacement therapy cannot pass into the offspring, whereas the transgenesis refers to the transfer of genes into fertilized ovum, which can be found in somatic as well as the germ cells and passed on to the successive generations. So here, the injection of a gene for growth hormone into a fertilized mouse egg gave rise to a giant mouse, the left one, and about twice the weight of his sibling, which is right. So we can also produce the giant animals with the greater genetic potentials, which is impossible by the natural method. And another thing is the gene disruption or knockout genes. Knockout genes means a gene's function can also be proved by inactivating the gene, then also looking for the adopted technology by adopted technology by the total genetic therapy is also done here. Then the specific genes can be targeted if their nucleotide sequence is already known, and hence here the gene disruption here the gene disruption or what we can say is sections of a muscle from normal gene disrupted mice as viewed under the light microscope. Muscles do not develop properly in mice having both myogenin gene if it is disrupted. Then manufacture of proteins or the hormones can also be done by genetic engineering technology. So the use of this recombinant protein for the production of human growth hormone, then insulin, interferons, and blood clotting factors. All these are done by this genetic engineering. Another approach for producing proteins via recombinant DNA technology is to introduce the desired gene into the genome of animal, engineered in such a way that the protein is secreted in animal's milk, facilitating the harvesting. So here you can also see manufacture of protein or hormones can also be done, especially from the production of the insulin from the pancreas, so that mammalian proinsulin mRNA is incorporated. Then using the reverse transcriptase enzyme, proinsulin cDNA is produced. Then it is joined to form a plasmid. Then recombinant plasmid is prepared here. Then the infected with E. coli. This type of the recombinant plasmid is infected to E. coli, for example, here. So it will direct the messenger RNA of the bacteria E. coli to produce the insulin. Here you can see. So it is a transformed bacteria. So entire bacteria will be filled with the insulin itself. So that bacterial promoter site, then eukaryotic DNA insert is will be done. Then it will be transformed into the E. coli. Then colony producing protein of the interest will be done. Then transferred colonies to replica plate lyse bacteria to expose protein. Then transferred protein into the nitrocellulose sheet. Then later these are added to radio labeled antibody specific for protein of the interest. Then autoradiogram is prepared, and hence manufacturing of this tissue plasminogen activating factor, which is administered to a patient after a heart attack and is made large quantities of mammalian cell. And new pharmacology using proteins produced by recombinant DNA technology as drugs is beginning to significantly alter the factors of the medicine. So here you can see that so many type of the medicines can be used with the recombinant DNA technology. So once again, site-specific mutagenesis can also be made as deletions of some genes, additions or insertions, and substitution. So ultimately, the required type of the genetic material will be produced. So the mismatched nucleotide will be inserted into a primer containing mismatched nucleotide is used to produce a desired change in the DNA sequence. Then the specific mutagenesis can also be inserted in a particular type of the plasmid. Then the designer genes are used for designer genes means specific type of the genes. So these novel proteins can also be created by splicing together gene sometimes that encode domains that are not associated with the nature. For example, a gene for antibody can be joined to a gene for toxin produces a chimeric protein that kills the cells that are recognized by the particular type of the genes is concerned. Hmm. So here the designer genes can be uh, incorporated into the required type of the genes, and so that it should be made very equivalent to that of the immunotoxins are being evaluated as anti-cancer agents. And designer genes are the one entirely new genes, and they can also be used against hepatitis B virus produced in the yeast cell is providing to be a effective vaccine against this deliberating viral disease. Then diagnostic aspect is also another thing. So ELISA The antibody test uses a recombinant HIV protein test for the diagnosis of HIV, and the DNA test detects the presence of HIV genetic material using RT-PCR or reverse transcriptase polymerase chain reaction. And development of RT-PCR test was made possible by the molecular cloning and sequence analysis by HIV genome. And diagnosis of the molecular diseases can also be done, like the sickle cell anemia, thalassemia, familial hypercholesteremia, cystic fibrosis, and many more diseases. So another thing is the prenatal diagnosis, the diseases where the genetic defect is known and a specific probe is available. Then prenatal diagnosis can also be done using the amniotic fluid synthesis, uh, especially from placenta, then also the uterus womb. So DNA cells are collected from as little as 10 ml of amniotic fluid by chorionic villi biopsy that can be analyzed by southern blot transfer. So, prenatal disease diagnosis can be easily done by this recombinant DNA technology itself. So, the early diseases very early in nature, can be easily diagnosed by using such type of the recombinant DNA technology. Then application of forensic medicine is also another thing to suspect the crime scene, and the advance in genetic engineering have greatly helped, specifically the criminals, to settle the disputes, especially the parenthood of the children. So, based on the basis of restriction fragment length polymorphism, the identity of a person can be easily diagnosed nowadays by matching with that of the mother and the DNA of a child, mother, child, and father one and father two, whenever there is a dispute is there. So then here you can see that the crime scene is there. Then the suspect 1, suspect 2, suspect 3 can be matched with that of the a child. Then which matches highest is identified as the father of the particular thing. Agriculture, of course, good quality food, etc., can also be manufactured by using the recombinant DNA technology. It is also one of the important technology in which the diagnostic materials can also be used, as NIF genes to cereals has given higher yield when compared to the other crops. Then also the apples, then industrial application like the enzymes synthesized by this technology are used to produce sugars, cheese, detergents, and many more things, and certain protein products produced by. And ethylene glycol or propylene glycol is one of the important thing from the genetic engineering technology. And animal cloning itself is once upon a time the dolly, one of the repeated terminology. So this has taken in such a way that the mammary cell donor and egg cell donors, they have taken these cells, cultured mammary cells are semi-starved, arresting the cell cycle and causing dedifferentiation. Then egg cell from the ovary is fused together with that of the cultured mammary cells. Then nucleus from mammary cell grown in culture, early embryo, then implanted uterus of a third sheep, then a surrogate mother, then embryonic development has happened, then the lamb genetically identical to that of the mammary cell donor. Biowar is another threat, of course, the virus is a threat. Then industrial golden rice can be produced using this particular technology is concerned. Recombinant technology will help in the production of chemical compounds of commercial importance, improvement in existing fermentation process, and production of the protein from the waste. Then human growth hormones can also be prepared by the genetic engineering technology using recombinant DNA technology. So, the human growth hormone is used for the treatment of the dwarfism. So, a human cell, a specific gene for growth hormone is isolated, then incorporated into the sticky ends of a DNA. Then later, the recombinant DNA is prepared for human growth hormone. Later, it is inserted into the bacterial cell for containing gene for human growth hormone. And such type of this bacteria will produce lot of growth hormone without any cadaver or any other. Then recombinant DNA technology is also used as a series of procedures for certain conditions like a molecule can enter a cell and replicate. So a plasmid donor and the gene donor. So isolation of the genes and cut bacterial plasmids are joined together to form with DNA ligase binding and then recombinant DNA molecule will be produced. Then human gene cloning is another thing where human DNA is isolated. It is inserted into the bacterium by restriction endonucleases, cleavage of the DNA, the insulin gene responsible for the production of insulin gene is going to be taken out and seals the human gene and plasmid. And ultimately, it is inserted into the bacterial cell and sufficient quantity of the recombinant DNA molecule is produced. And of course, now the lot of demand is for the production of the antibiotics. So they are the chemical substances produced by both microorganisms synthetically. So they can inhibit the growth of bacteria and other microorganisms and even destroy them. Penicillin is the first antibiotic discovered by Alexander Fleming in 1928 using the recombinant DNA technology. Then there is a one step called as the production of human insulin. A bacterium like E. coli is inserted with the plasmid of the gene containing the insulin of the human being, and in the step 1 the bacterium is inserted with that of the plasmid with the required DNA, then the plasmid outlay enzyme, then enzyme with that of the plasmid wherein with the human insulin gene is inserted in the step 3, whereas the genetically engineered material is once again incorporated into a new bacterial cell. Then later the bacterium divides and begins producing lot of insulin because the bacterial characteristic thing is they go on dividing into several type of the victim. Then vaccines are also produced like the hepatitis B, foot and mouth disease, where vaccines, especially if we come to the animal side, the foot and mouth disease vaccine is already produced using the recombinant DNA technology. Then gene therapy is another thing wherein which the hemophilia with normal genes can be treated because the abnormal genes are removed out and the normal gene is being replaced in certain genetic disorders. The defective protein can be simply replaced with that of the normal protein in the cell. Agriculture also the new type of the plant biologically engineered BT cotton and many more, but they are having lot of opposing from the people. It has its against the nature. Then all these things, production of the enzyme, production of the transgenic animals, like other thing is the HCG, human chorionic gonadotrophin. It is a therapeutic agent prepared from the human proteins in their milk. Fish like common carp, catfish, salmon, and goldfish contain human growth hormone. In animal husbandry and sericulture, it is used for the enhancement of the milk production. Then the animals are produced in such a way that they are going to produce very high type quality of the milk. And in case of birds also, the egg quality and meat production quality in the respective animals. And in summary, recombinant DNA technology, it can be used in case of the medicine, agriculture, industry, research, diagnostics, then for the treatment aspect, preventive aspect, better and different yield of a crop, maybe in case of the animal or the horticulture side. In industry also, a lot of detergents, then additives, sugars are being added. Then infection and metabolic infections. So gene mapping uh, is also used for the crime detection, or if it is not found also we can save the innocent people. In crime, it's a difficult task to commit nowadays because it should not never be. Then finally the vaccine is produced. Then thank you very much for being with me for a long